Assalamu alaikum and Bismillahir Rahmanir Rahim. Dear students and we are here with a new, very interesting and very important topic. And the topic is basically CBC, and I have given the name ABC of CBC because in this topic we will discuss all the parameters which are included in CBC. And what is CBC? Is a complete blood count, or it's also known as full blood count, that is called FBC, and it is a blood test that used to evaluate your overall health and detect a wide range of disorder that may be anemia, infection, leukemia. And a complete blood count test measures several components and features of your body, including red blood cells, white blood cells, hemoglobin, hematocrit, platelets, and so on. Abnormal increase or decrease in cell count, as revealed in a complete blood count, may indicate that you have an underlying medical condition that call for further evaluation. So let's start the topic, and this topic is for both fourth and sixth semester. My name is Tanveer Tara, and I am lecturer at Sarhad University of Science and Information Technology. Now, what is the significance of CBC? Basically, we have three components in CBC, main three components. One is RBC, red blood cells, and the second one is white blood cells, and the third one is platelets. And the CBC test is useful in diagnosis of various type of anemia, that we are looking hemoglobin, and whenever we find that hemoglobin level is decreased, so we find out that there is anemia, and which type of anemia that is we will discuss in further slides. Similarly, we have WBC. If WBC count is high, then we will think about that is there any infection, bacterial infection, viral infection, or some allergic condition. And if there is platelets count high or low, then we think about the clotting problem. Maybe there is thrombocytopenia, or there may be thrombocytosis. So these are the components of CBC, and in hematology we call them parameters of CBC. So these are red blood cells, hematocrit, hemoglobin, MCV, that is mean corpuscular volume, mean corpuscular hemoglobin concentration, that is MCHC, red cell distribution weight, RDW, and white blood cell WBCs, DLC, differential leukocyte count, absolute count, then platelet count, MPV, mean platelet volume, PDW, platelet distribution weight, and we have one another, that is PCT, platelet crit. The first parameter is RBC, red blood cell, and it's red blood cell count basically. Red blood cell count is usually carried out as a part of full blood count, and women usually have a lower RBC count, that is 4 million to 5 million cells per microliter, and the men have 4.5 million to 5.5 million cells per microliter. Both are different, and sometimes we have the report like they have given the range, and that is 4.5 to 6.5 million cells per microliter. This is general range both for male and female. Now, if RBC count is lower, then we think about anemias, and how we can approach anemia we will discuss in the further slides. But if RBC count are higher than the normal, then we think about the erythrocytosis. And the best we describe erythrocytosis is polycythemia, because polycythemia is sometimes called erythrocytosis, but these terms are not synonyms, because polycythemia refer to an increase in red blood cells. So whenever RBC count increase, then it's called polycythemia or erythrocytosis, and whenever we see that red blood cell count are decreased, then we think about the anemia. The most important parameter in CBC is hemoglobin, and on the basis of hemoglobin, we can say that the person is suffering from anemia. What is anemia? Anemia is the low level of hemoglobin according to age and sex, and when the hemoglobin level fall from its lower level, then it's called anemia. For male, its normal range is 13 to 17 gram per deciliter, and for female, it's 12 to 16 gram per deciliter. And hemoglobin is the first parameter we look for, and on the basis of hemoglobin, we go for further indices. Like when we see that hemoglobin is low, then we look for MCV and MCH and MCHC, that whether anemia is microcytic, macrocytic, or normocytic. Hematocrit, or it's also called PCV, packed cell volume. It is the ratio of the volume of red cells to the volume of whole blood. And we can say that the hematocrit is the proportion by volume of the blood that consists of red blood cells. The hematocrit is expressed as percentage. For example, a hematocrit of 25% mean that there are 25 milliliters of red blood cells and 100 milliliters of blood. And hematocrit or PCV is the percent of blood volume filled by the erythrocyte, and thus a measure of oxygen carrying capacity of the blood. We can say in another example that if there are 100 ml of blood with the HCT of 45%, the erythrocyte acquire 45 ml. MCV, mean corpuscular volume. It is the most important parameters after hemoglobin, and when we see that hemoglobin is low, then we think about the MCV, that whether anemia is microcytic, macrocytic, or normocytic. The normal value for MCV is 80 to 100 femtoliter, and whenever it reduces from 80, then it's called microcytic. And if it more than 100 femtoliter, then it's called macrocytic. The cells are larger in size, and if it is a normal range, then it's called normal. So the formula for mean corpuscular volume is hematocrit multiplied by 10 divided by RBC. This is a manual formula. We can multiply hematocrit with 10 and then divided by RBC. So mean corpuscular volume is the average volume of red blood cells, and it can be directly measured by automated hematology analyzer as well, and by the formula as shown here. MCH, mean corpuscular hemoglobin. MCH levels refer to the average amount of hemoglobin found in the red blood cell in the body, and MCH value refer to the average quantity of hemoglobin that is present in a single red blood cell. And on the basis of MCH, we can say that anemia is hypochromic or normochromic. The normal value of MCH is 27 to 32 picogram. Like from MCV and MCH, now we can say that if we have hemoglobin and that is lower than its normal, then we look for MCV, and that is also lower than 80 femtoliter, and then we have MCH that is lower. So we can say that the cell size is lower, so anemia is microcytic, and its MCH is lower, it's hypochromic, so anemia become microcytic hypochromic anemia. MCV, MCH, and MCHC are combinedly called red blood cell indices. And the third one is MCHC, a mean corpuscular hemoglobin concentration. Its normal value is 26 to 34%, and whenever it decreases, then we can say that it is hypochromic, and if it's normal, then say that it's normochromic. It also can be calculated by the formula, like whenever we multiply hemoglobin with 100 and divide it by HCT or PCV, we will finally get MCHC. It is a measure of the average concentration of hemoglobin inside a single red blood cell. RDW, or red cell distribution width. It is an important supporting parameter. Red cell distribution width helps measure variation in red blood cell volume and its size. Mean it it's correlate with the degree of anisocytosis. Anisocytosis means variation in size of red blood cells. And normal red blood cell maintain a standard size that you all know it's 7 to 8 micron in diameter. And if your RDW is elevated and if the range of size is large, this means that if on average your red blood cells are small but you also have a lot of very small cells, your RDW will be elevated. Similarly, if on average your red blood cells are large but you also have a lot of very large cells, your RDW will be still elevated. For this reason, RDW is not used in isolated parameters when interpreting a complete blood count. That's why in the start I said that it's a supporting parameter. It provides shades of meaning in the context of the hemoglobin and mean corpuscular volume. And if your RDW is too high, it could be an indication of a nutritional deficiency, such as deficiency of iron, folate, or vitamin B12. These results could also indicate macrocytic anemia as well. The normal range for RDW is 10 to 15%. This is an important picture. So after discussing red blood cell, hemoglobin, PCV, then MCV, MCH, MCHC, and finally we'll get RDW. These are important indicator or these are important diagnostic tool for identification of any disease like anemia or it might be macrocytic, microcytic, or it may be a normochromic normocytic anemia. On the basis of these parameters, we can easily understand which sort of anemia we have. Then we have WBCs, and on WBCs I have my lecture that is DLC, that is separate lecture. So two parameters, most important parameters, RBC and WBC. Now the third one is platelet, and platelet are basically tiny blood cells that help your body from clots to stop bleeding. If one of your blood vessels get damaged, it sends out signal to the platelet, and the platelets come over there and make and aggregate. That is functional. Normal platelet count range from 1,50,000 to 4,50,000 per microliter, and if the count is more than 4,50,000, then the platelet condition is called thrombocytosis. And if this range decrease from 1,50,000, then it's called thrombocytopenia. And there are certain diseases in which we have thrombocytosis or which we have thrombocytopenia. Similarly, mean corpuscular volume, we have mean platelet volume, and it's called MPV. Its normal value is 9.4 to 12.3 femtoliter, and the normal diameter of a platelet is 2.65 to 2.9 micron. MPV, or mean platelet volume, is a machine calculated measurement of the average size of platelets found in the blood and is typically included in blood tests. Now it's become a common part of CBC. In each and every report you will get MPV. Mean platelet volume is the average apparent volume of all the particles in a blood sample that are counted as an individual platelet. And abnormally low MPV values correlate with thrombocytopenia, or sometimes we get it in aplastic anemias as well. PDW, platelet distribution weight, and it's also the same indicator as for the red blood cell distribution weight, that is RDW. So the normal range for PDW is 10 to 17.9%. And basically PDW, platelet distribution width, reflects how uniform the platelets are in size, and normal PDW indicates platelets that are mostly of the same size, while high PDW means that the platelet size varies greatly. PCT, platelet crit, and platelet crit is basically the volume occupied by platelet in the blood as percentage. and it's also calculated by a formula, that is platelet count and multiplied with mean platelet volume, then divided by 10. The normal range for a platelet crit is 0.22 to 0.24%. So it's all about the CBC parameters, and we have three basic man parameters, that is WBC, RBC, and then we have platelet. For WBC we have differential count, absolute count, and we also have discussed it in a very detail in our DLC lecture. That's why we do not discuss it over here. Then we have the second parameter, that is RBC, and RBC have hemoglobin, then MCV, MCH, MCHC, and RDW. These are the most important parameters. On the basis of hemoglobin, we can say that the person is suffering from anemia. Then on the basis of MCV, we can say that it's microcytic, macrocytic, or normocytic. And then we go for the word hyperchromia on the basis of MCH and hemoglobin concentration. So these all are about the parameter, and the finally we have platelet, that while looking the platelet count, we can say that person suffering from thrombocytopenia, or it might be thrombocytosis. And then we have also basic parameter for platelet, like platelet grit and mean platelet volume, and also we have PDW, that is platelet distribution width. So it's all about for today, and inshallah we will see you in the next lecture, that will be in